Welcome again to the Gable Hall channel. Today I will be talking to you about plant responses. And this experiment is a required practical for triple science biology. We're looking at cress seeds especially, and we will be changing the environments in which the cress seeds grow. So the first, we'll place the cress seeds directly under a light source so that there's light coming directly onto the cress seeds. We count 10 cress seeds, place it in a petri dish lined by filter paper, and then the next we'll place the same number of cress seeds, 10 cress seeds, in a petri dish again lined by filter paper soaked in water. This, as you can see from this, the light source is not directly over the cress seeds. The light source is coming from the side. Okay, so light is coming from the side towards the cress seeds. Then we place a third set of cress seeds, 10 cress seeds again, within filter paper soaked in water in a petri dish. We're going to leave this under the same conditions, so in a dark room, and leave it for a couple of days. And then after those days, we'll come and look at the growth of the cress seeds. After those, let's say about 10 days, we'll realize that the cress seeds under the direct light source will grow vertically and they wouldn't take any direction. The cress seeds from this petri dish will grow towards the light. The cress seeds from this petri dish will also grow towards the light source. The question is, why do cress seeds actually grow towards the light source? That is a question. The answer is very simple. The growth of plants is controlled by hormones called auxins. Auxins control how fast plants grow. So for example, in shoots, auxins will cause the plant to grow faster. So when you expose, let's say this is a plant growing up, and then light is coming from that direction, the auxins will move towards the shaded side of the plant, and it will cause that shaded side to grow faster, and so cause it to bend towards the light. Okay? Plants use that as a form of adaptation so that they are always receiving enough light for growth. We know that light is necessary for photosynthesis, and that is why plants want to always grow towards the light. So this, the cress seeds here will definitely grow and fold towards the light in order to obtain more light. And as I've explained, it's because as the light is coming from this direction, the auxins will move towards the shaded side and cause that side to grow faster than the side that is facing the light, and so the plant will bend that way towards the light. The auxins from this one will also grow towards the light for the same reason. The light will come from that direction, and so the auxins will move to this side, and when they move to that side, they will now cause the plant, this side, to grow faster, and so the shoot will bend towards the light. In this experiment, you may be asked about a couple of things about scientific skills. One of the key things they might ask you is, what did you change in the experiment? What was the independent variable in the experiment? The independent variable is the direction from which we bring in the light. There's light above the cress seeds, there's light from the sides. What is the dependent variable? The dependent variable will be the direction in which the cress seeds actually grow. Do they grow away from the light? Do they grow towards the light? That is what we are measuring, that is what we are observing. Another question they might ask you in this experiment is, what are the control variables? What should we and why do we need the control variables? The control variables are the things that will affect the growth of a plant. So we need to keep the same amount of water in the petri dish. We need to keep the temperature the same. We need to keep the lighting the same. As you know, photosynthesis is affected by the amount of light. We are not investigating the amount of light in this case, we are investigating the direction of the light. So we need to make sure the light is the same. We need to make sure the temperature is the same, because if we change the temperature, it's going to affect the enzymes, and so it might affect the growth of the plant. So amount of water, light intensity, the temperature, the type of seeds, as well as the number of seeds. If you realize, we use 10 crest seeds in each one. Also, you can be asked on how you can improve the experiment. What can you do to improve this experiment? Obviously, we can improve the experiment by repeating the same process. We might have more than one petri dish and more than containing 10 crest seeds each. Then when we get the results, we can find the mean by eliminating the anomalous result. In that way, we'll have a more accurate result, which makes it more valid. We hope this video will help you, and I wish you the very best in your exams.